So the next one we're going to be taking a look at is actually a mutation. So bioinformatics can show us mutations in our sequences. So the receptors CCR5 and CD4 are proteins normally found in the surfaces of T cells that form part of our immune system. When a person becomes infected with HIV, the virus attempts to dock on the surface of the T cell using two receptors. After docking, it then infects the cell and replicates. Over time, the virus also destroys them. Now, this destruction to the immune system over time can eventually lead to AIDS and eventual death. However, there has been a mutation found in the CCR gene that makes people who are homozygous for the gene very unlikely to become infected with HIV. Those who are heterozygous are more likely to become infected with HIV than those who are homozygous, but their disease progression is much slower than those without mutation. It is estimated that 1% of people who descended from Northern Europeans are homozygous for this gene, and a further 10 to 15% are heterozygous. This mutation is called CCR delta 32, as it involves the deletion of 32 base pairs on the gene. This causes a frame shift which results in a premature stop codon, and this means the protein is much smaller than the normal version and so no longer sits on the cell surface. So below is a representation of the normal CCR5 protein, and you can see that the protein is in orange and the plasma membrane in grey and red, and the bits of this protein outside the cell are going to be the receptors. So we're going to take a look at the delta 32 mutation and compare it to the normal CCR mutation using bioinformatical tools, and we'll also be using the PDB, the protein data bank, to take a look at the CCR protein that has been modelled. So what we're going to do is we've got our sequence here and we've got coding normal and I've called it coding mutant. So we're going to highlight this sequence and we're going to pop it into a program called Clustal Omega. Now this Clustal Omega program is going to align our sequences. So make sure protein's in the box, paste it in and press submit. And again, depending on where in the world you are, this may take a while or this may be very quick. Okay, so we have our alignment here, and so far we found no changes until we hit here, and here we have a problem. So here you can see that we have a gap. Now all the rest is absolutely fine. And these are two human CCR5 receptors. Now this gap, and I've counted, and these are 32 base pairs long. So this is a 32 base pair deletion. And that is what we're seeing here. We've got the normal one and we've got the mutant one, and it's all the same up until we reach the mutant protein. Now this 32 base pair mutant actually causes a frame shift error, and it causes a premature stop codon. Now we've got a choice. Either we can search for this ourselves by starting at the very beginning and checking our base frames and going from codon to codon to codon, but we're not going to do that. This is why bioinformatics invented bioinformatics tools, to stop us doing this. So what we're going to go ahead and do is we're going to pop it into emboss transseq and we're going to have a look. It's going to translate our nucleotide sequences and it's going to translate them into protein. And what it does is it finds a stop codon, highlights it with a star. So we're going to look and see if we can find the first stop codon. So what you need to do is copy the same sequences into emboss transseq and press go. Okay, so we have our result here. So coding normal begins with a methionine, like every good protein does, and it ends with a stop codon, which is fantastic. Now the mutant also ends with a methionine, but where's its stop codon? Now what's worrying is the first stop codon is in the middle of the protein, and that's here. So the first stop codon, because of the frame shift error caused by the 32 base pair deletion, we have a premature stop. So all of this sequence after here doesn't matter. It's no longer translated. It's no longer transcribed either. So it doesn't matter. So this results in a very small truncated protein that doesn't sit in the cell surface correctly. Now this no longer functions as a receptor. So this is why this mutation makes people very resistant to HIV. Now not immune, there have been cases where people have got HIV, but it does make them very resistant. So the last thing we're going to do is we're going to take a look in the protein data bank at the CCR5 protein. So that's for MBS. And this is the crystal structure of the CCR5 chemokine receptor. Now there are two of them, and they're two protein subunits, and you can move along and play with them using the 3D viewers. So you can actually begin to play with the protein, you can rotate it, you can change the settings. So we can have a look at cartoon or secondary structure. And obviously my laptop's being a little bit slower, but I'll leave you to play with that. And what we can also do is we can actually hover over the protein and see the amino acids involved. So if we have a look here, it'll tell you that that's a leucine right there, and you can kind of stroll across and you can have a look at the amino acids involved and that kind of thing. Now this isn't the mutant one, this is the healthy one. I couldn't find one of the mutant ones, so we can't really take a look. So there we have it, guys. So enjoy.